Watch the attached WHO hand washing demonstration video and identify the correct sequence of hand washing steps shown. Okay. Because they have shown 2 or 3 steps. Okay. Okay. Somebody has got the answer. Yes. Good. Very good. Very good. Okay, guys. Very good. So hand washing, basically WHO tells around 10 steps. So what are these steps? So first, wet your hands with water. First step. Then apply enough soap. Then palm to palm. Okay. First, hands with water, enough soap, then palm to palm. So after palm to palm, right palm over left dorsum and left palm over right dorsum. Soap, water, soap, palm to palm, then palm over the dorsum. Next step, palm to palm with fingers interlaced. Next, back of the fingers. So first, wet hands with water. So this procedure takes around 40 to 60 seconds. Apply enough soap to cover all the surfaces. Rub hands palm to palm. After palm to palm, then palm over the dorsum. So this is the palm, this is the dorsum. Palm over the dorsum. Then palm to palm with the fingers interlaced. Then back of the fingers. Then next we are going to the thumb. Right thumb, left thumb. So next is clasped fingers. That is the fingertips. This is only what they are meaning, the fingertips. The clasped fingers are the fingertips. Then we are washing dry with a single-use clean towel. Then while closing the tap, you can use your elbow to close the tap, or you can use that towel itself to close the tap. So you should not touch the tap again with your clean hands. Okay. So this is the steps. So wet your hands with water. Enough soap. Rub the hands palm to palm. Right palm over left dorsum and left palm over right dorsum. Then palm to palm with fingers interlaced. Okay, now we will stop. So next question. So watch the attached video on biomedical waste segregation and identify the correct color-coded disposal for the biomedical waste items shown. See, the one by one, first is red color bag, then some culture tubes, then some plates, media. So what is the biomedical waste bag? You will put all these things. Okay. Good, doctor. Good. So it is A. So it is a yellow, dirty fellow. It's yellow, dirty fellow. So what did you see here? So it is a red color bag. All the biomedical waste bags, all the culture plates. Then why the glass? Cytotoxic. This is the important thing. Cytotoxic. Anything which is cytotoxic also will come under the red color. Cytotoxic. Even if it is a glass vial, if it is something which can affect the genes, that is a cytotoxic glass vial, they will also come under yellow color. So what are the things in the yellow? So we have human anatomical waste, animal anatomical waste, soiled waste like a cotton with a blood, expired or discarded medicine, chemical waste, then any linen which is contaminated with blood and the body fluids, microbiology waste, biotechnology waste, as well as the clinical lab waste. So all of this will go into yellow colored chlorinated plastic bag. Main type of disposal is incineration, heating at high temperatures. So next, now we are going to watch the attached video and identify the type of the mosquito which lays this specific type of eggs. Okay, which mosquito lays this specific type of eggs? Yes, good. Why it is C for Culex? Culex, it is for clusters. So Culex, it lays the eggs in clusters, like a grapes. So like grapes, the Culex, C for C, Culex is clusters. Okay, very good, doctor. This is Culex is clusters. So Anopheles, you will see another video with for the X. See Anopheles, you will have a lateral floats. If you are seeing this lateral floats, then it is Anopheles. Anopheles is single. It is a Morata single. It is a single boy or a single girl. Single Anopheles is single. Aedes is also single, but Aedes it is a this type of shape is called a cigar shape. But if it is having lateral floats, it is called as the boat shape. This is a boat shape. It is boat shaped and it has lateral floats. It's like a boat with a lateral float. But Q like C for C, it is alone will be in clusters. Q will be in clusters. So this is the difference between the X. Then coming to the larva, Sonophilus, the larva rests parallel to the water surface. There is no siphon tube. But here there will be a siphon tube. For Aedes and Culex, there will be a siphon tube. Okay, next, we're going to watch the video and identify the type of mosquito breeding on this specific water source. Okay, which type of mosquito breeds on this water source? Okay, yes, good. Okay, very good. I think you are all up to it. Okay, nice. Okay, this is what? Artificial collection of water, A for A. Remember, A for A, artificial collection. What is this artificial collection? Any tire. You are having a tire. In the tire, water is there. Then you are having a pot. You are growing the trees in the pot. In this, water is accumulating. So any artificial collection of water, it is like a ditch type of thing, like what is the chalk like a sewage. In those areas will be culex. So ditch, very like bad smell areas where the water accumulates, there will be culex. But Aedes is mainly in the artificial collections of water, mainly like the whatever happens around our houses after a rain. So after rain, we have to mainly look for Aedes. So only mainly after rain, We are looking for like dengue awareness program. We are looking the dengue has spread. 3 children died of dengue. Mainly it happens after rains. So we are having a window. In the window there will be a slab. There can be water can accumulate. Okay. So breeding habit, Anopheles, clean water larva. It was created using AI, doctor. So do not worry. So the video is mainly about this cup. Okay. So do not worry about the larva for that video. Okay. So clean. So clean water. So Anopheles is clean water. Anopheles is clean water. Since he asked about larva, so this is the larva. Okay. So there we do not have a proper clarity for larva. If you see the Anopheles larva, if it is the water, inside the water, Anopheles larva will be clinging like this with the whole body. But the Aedes and the Culex larva will be like this. There will be a siphon tube like a teeth. There will be a siphon tube for the larva. Next, breeding habit, clean water. For Culex, it is a dirty, polluted, or a stagnant water like a chakra or a sewage water. Aid is clean water, but artificial collections. Mainly, Anopheles you can see mainly in paddy fields. Clean water, paddy fields, it can be Anopheles. Identification point. So, Aedes, you all know the black and white, the tiger mosquito. The black alternate black and white bands. Anopheles, it is always sitting with a lot of Anopheles has a lot of ego. So its back will not touch the ground. So it will sit at 45 degrees. It has a lot of ego, Anopheles. So it is sitting at an angle of 45 degrees to the surface. And if you see the wings of the Anopheles, that is mainly the veins. The veins will have the spotted wing appearance. It is due to the veins of the Anopheles. Okay. Next question. See, look at the picture carefully. Zoom, try to zoom and see the egg. Okay. See, single, it is single, but try to zoom and see. Zoom. Nicely zoom and see. Okay, this is the exact picture. I took from some YouTube video. So in this you can see. Nicely zoom and see. See the what type of egg. See the type of the egg. It has 2 lateral floats. Yes, yes. So, yes, there maybe you may get confused because of the lack of the clarity, but when you zoom in, you can see the 2 lateral floats. The 2 lateral floats are there. So, because of this lateral floats, it is Anopheles. Because of this lateral floats, it is Anopheles. If you see mainly, someone told Mansonites. So, how will be Mansonites means like there is a water plant or aquatic plant. The eggs will be attached to the aquatic plant like this, like a star-shaped clusters. It will be mainly attached to the roots. So it is not Mansonites. So Culex cluster. So it should be either Aedes or Anopheles. Now when you are zooming and seeing, you can see this boat shape. So it may not be so much visible for you, but there are visible in some of the eggs it is visible. So boat shape. So only it is Anopheles. Okay. You can see. So if it having boat shape eggs, it will be Anopheles. A boat shape. A boat with a lateral floats. So what is the center? Center you all know. What is the center? The center is Culex. Good. The center is Culex. So what is to the rightmost or the rightmost part of your screen? It's the Aedes. Aedes is cigar shape. Cigar. Okay. So okay, watch the video and identify the apparatus. You'll easily identify. So actually all the cups will be some white because of that two mux has come. Don't worry. So it is a Horax apparatus. How can you identify this Horax apparatus? You saw a box. When you open this box, you will generally see the 6 white cups. Then you will see a indicator solution. A indicator solution, which is helpful for turning the solution into blue. So we will pour the water in all the 6 cups and we'll put the indicator solution, 1 drop in first cup, 2 drop in second cup, 3 drops in third cup, 4 drops in fourth cup, and go on. So we'll see which cup is turning into blue color first. There are 6 white cups and an indicator solution. The amount of bleaching powder required. Estimate the chlorine demand of the water. So what is the use of the Horax apparatus? I can estimate the chlorine demand of water. So for example, now we can take the third cup. The third cup is changing in color. That means dash into 2 grams. So 3 2s are 6. So 6 grams of bleaching powder are required to disinfect 455 liters of water. This is like a constant for 455 liters dashed into 2 grams. Third cup means 6 grams. Fourth cup means 8 grams. About Horax. Next, watch the video and identify the apparatus used. So the apparatus has 2 tubes. One tube is white in color and another tube is somewhat yellow in color. So sometimes instead of this tube, they can also use a color paper. Like this color paper can be there. This color paper will tell you. So this is 0.1, 0.2, 0.3, 0.5 parts per million. What is this? What is the amount of free chlorine or residual chlorine? Yes, very good, doctors. It is the fluoroscope. To identify the amount of free chlorine or residual chlorine, that is the fluoroscope. That is the fluoroscope. It is like in physiology, have you studied that hemoglobin salicylic method, that hemoglobin apparatus, similar to that in our community medicine, this is the fluoroscope. So fluoroscope is a portable apparatus, visual color comparator. We are comparing the residual chlorine levels in drinking water and other types of water bodies by adding a chemical reagent. Usually we add orthotolidine or orthotolidine arsenite or other things to the water sample, and then we can identify in orthotolidine arsenite test, we can calculate the free chlorine plus residual chlorine separately. So we can tell how much is the amount of the free chlorine, or it is 0.5 parts per million, or it is 1. For swimming pools, it is higher. For normal drinking water, 0.5 is half. Okay. Now this is a image-based question. Okay. If you drink this water, if you are drinking this water, which disease you will get? And what will be the hardness of the water? What will be the hardness of the water and which disease you will get? This may come maybe like I thought of taking a video but did not have time, like pouring water from a bubble top. So what is the disease caused by this water? Yes, good. It can cause cardiovascular disease or a coronary water disease because of the deposition. It can lead to the it can lead to atherosclerosis. It leads to the deficiency of the ions like calcium and magnesium. It leads to low TDS. TDS means total dissolved solids. So that is also called as decreased hardness. So this is a machine which is even available in Amazon, Flipkart, and all you can see. This is called as a TDS meter. So it is like a thermometer. If you see some apparatus like thermometer, but they are putting it in water, you can see here the reading is 108. Okay, it is reading is 108. So what is the normal hardness level of drinking water? What is the normal hardness levels for drinking water? What is the normal hardness levels for the drinking water? Drinking water. Yes, moderate level. Yes, 1 to 3 milliequivalent or 50 to 150 mg. That is our 1 to 3 milliequivalents per liter. it is moderately hot. But when we are doing this process, it may become less than 50, it may become soft water. Okay, now we are going to see the video and identify the correct statement. So now all these are easy. Now this is somewhat you have to apply your brain. Apply your brain, take some few seconds. Which is the correct statement? Okay, computer, okay. You are sitting in front of your screen and you are seeing. See here, frozen, the sense means like which has been exposed to freezing temperatures and lost its efficacy. See, this is a shake test. In the shake test, always the center is the control vial. Control vial means something which we freeze it. We freeze it, we are freezing it, and we are keeping it. So it is a frozen control vial. Now we are having two vials which we have put inadvertently inside the fridge. And we have to see which has lost its efficacy, which is good. Which is like in which vaccine vial, what I told in the class, in which vaccine vial, sambar has become like a rasam, like your mess rasam, like mess sambar. In which vial the sambar has become like a rasam. See here, sedimentation. But here it is uniform, it is okay, good. But here everything got sedimented. हो गया मर गया खत्म मुड़न போச்சு. So it is sedimented. Sedimented means it has been exposed to freezing temperature and it has lost to efficacy. Okay, so the sambar has become like a rasam, so do not use this. So this has been exposed to the freezing temperature. So this has been exposed to the freezing temperature. So it has been frozen vial and it has been exposed to the freezing temperature. So this is a frozen control vial. Now this one is the good vial which we can use. Good vial, it has not lost its efficacy. Before it got frozen, we took it out and we have the hero has saved this vial. So the right vial is not frozen in the meaning the right vial is good to go. It has not lost its efficacy. So guys, you got it? Okay, you got it? Yes. Thank you. Right vial exposed. Good. Okay, good. So what is this shake test when we do? To check whether vaccines have been exposed to freezing temperature or not. A vial of frozen vaccine. No. Stop this. Okay, a vial of frozen vaccine after it has been thawed. So thawed means like after we have unnecessarily frozen it or unknowingly frozen it, we keep it in the normal temperature for around 30 minutes. That is called thawed. No longer has the appearance of cloudy liquid. It tends to form flakes that settle at the bottom. So the sambar becomes like a rasam, mess. So in mess, the sambar, if you go and eat late at 3 o'clock, it becomes like a rasam. You ask, Anna, rasam only you put today. Where is the sambar? Because everything has settled down. So it has been exposed to freezing temperatures. So it has lost its efficacy. We have to compare the rate of sedimentation in the control vial and test vial. If the rate of sedimentation is same in both vials or faster in the test vial, then the vaccine is balanced. Okay, control vial, test vial. So sedimentation, no, don't use it. Shake test failed. Cloudy, good. Shake test pass. Okay, cloudy. Okay, next. See the video fully and then answer. Do not hurry. Okay. Sorry. See, in the exams, there will be only video, no audio, because they will not give earphones. Okay. Now see and read the question clearly. Identify the color-coded disposal for the biomedical waste item. Now it is shown. Now what they are showing now, for this where you put. Yes. Yes, it is. They are not asking about the needle. They are asking about the syringe without the needle. So where you put the syringe without the needle. Yes, red. So immediately seeing, you should not go for needle means sharp. You have to see the picture or the video fully. Now the needle has gone inside that needle destroyer. So forget about the needle. Now you have to worry about only the plastic part of the syringe. So the plastic part where you put it in red color. Oh. So next, this is a easy. So in red color, I will just summarize that later also. Okay, now see the attached video. Identify the color coded disposal for the biomedical waste items shown. Yes, they are all human anatomical waste. It is yellow color, including even the dead fetus. Also, blood bag is also yellow color. Remember, blood bag. Many of you will have doubts. Blood bag is also yellow color. Any glass vial with cytotoxic. Glass vial alone means blue. Blood glass vial, it has cytotoxic drug. Then also it should come for yellow. Any discarded or expired medicine. These things you should not forget. So blood stained gloves, yes, blood vacuuminers, you can see it is more soiled. So there is also a category for a soiled waste. But plastics, we will avoid putting it in yellow because here we may incinerate. No, because of that, it may heat so much. So plastics, we will always go for red. Okay, plastics means red. Remember, plastics. But glass vial, if it is a cytotoxic drug only here. So normal vaccine vial, no need, only blue. Like a cancer drug, you are using some cancer drug. Blood vacuum after usage in labs. Okay, vacuum thinner, it is made of what? Plastic or like a glass? So you have to see, plastic means you always go for red. So plastic means always red. Only if it is having cytotoxic, then only yellow. So only glass, maybe even it can have some blood. It's not a problem. We can go for a blue. Broken also, but if it is cytotoxic, broken or not, if it is cytotoxic, yellow. But broken glass also blue. No need to put in white. Broken also, but if it is glass without any cytotoxic drug, it is blue. Without any cytotoxic drug, it is blue. Okay, here the correct term is which is plastic or glass. That is only matters. You can go around and round and ask in different ways. But what the item is made of, if it is plastic or not, if it is visibly soiled. But if it is plastic means never put in yellow. Okay, never put in yellow. Empty vaccine vial also blue. Blue. Vaccine vial, glass vial. Remember, glass vial, blue. So glass means blue. Glass means, the core idea is like this. Glass means blue. So plastic, red. Catheter, okay. Urinary catheter, it is red. Urine catheter, red. Urine catheter is plastic, red. So expired medicines is yellow. Both expired medicine also yellow. Okay, I will go there again. Expired medicine also yellow. So just see it again once. So vaccines always it is mainly blue. Vaccine glass vial blue. So yellow human anatomical waste, animal anatomical waste, soil waste, expired or discarded medicine, chemical waste, discarded linen, mattress and beddings which are contaminated with blood, body fluid, protein, mask and bone which are non-plastic, which are contaminated with blood. Microbiology, biotechnology, and the clinical lab waste, which may have some microbiological organisms, then it can go for yellow. So here mainly incineration means heating. So mainly we will avoid heating of the plastics. So if there is plastic, we will not go. So only incineration. So only we have a separate item for plastic. It is mainly red color. So red color is mainly for plastic recyclable. So all the IV tubes, IV bottles, IV sets. So I'm not able to get what is vacuum cleaner made of, whether it is glass or plastic. I want that detail. Then only I can tell. Plastic means red. Plastic means red. Like if it is a glass means we can go for blue. But microbe, since the microbe is there, like plastic here culture. So culture plates is coming in yellow. So culture plates are coming in yellow. Even it can be made of plastics, but they are putting as culture plates. They are putting it as in yellow. So I think maybe vacuum trainers also, since like blood black, vacuum trainers may also come in yellow. But I need some confirmation. Maybe from micro we can get that. So rails tube, catheters, gloves, urine bags, syringes without fluid is contaminated or not. Even if there is blood in the urine bag, even if there is blood in the IV set, it is red. Even if there is blood, it's okay. So these are all mainly red color, non-fluorinated. So the plastic waste also should never be buried. See here. So here, yellow burial is one type of method. So only we show they are differentiating. So plastic waste should never be sent to the landfill sites. These things should never be buried also. They will not heat it also. They will not heat it also. Then white color waste shops, including the metals like our needles. So first two things are bag. From here it is box. So from here it is box. So blue color is also it is box mainly for glassware and metallic body implants. But in case if there is any cytotoxic drug, we will not put in blue color. Instead of the blue color, even though it is a glass, we'll put it only in yellow. I think the hand washing step. So the video got cut, just show the hand washing step 1 minute. I'm not sure, doctor. Black bin is not there, but maybe I will check and tell you whether all these things will change when there is thing X-ray film, fluid, HIV. Okay, radiological. They are telling radiological. We have to treat it separately, but anyway, I will also check and tell you the correctly answer. So you want HIV hepatitis B? Okay, we'll take a photo and I will tell you distinctions. These questions, I will confirm and tell you, because if I tell something and if it goes wrong, it's fine. I'll confirm 2 or 3 times and then I'll tell you.